Hi friends, after a long time I am doing this video. Uh, Today this is not a normal video. This is a challenge video for me. Most people uh, struggle to remember even 50 genetic terms. It is typical one. Once I am asked myself, um, what if I tried 1,000 genetic terms? If I fail, you will see it. If I succeeded, it changes everything. Okay, first, how many of them I recall genetic terms? Okay, let's see. First, gene construct, genome refactoring, genome integration, genome knockout library, genome attribution system, gene staking, gene permitting, genome minimization, minimal gene set, genome transplantation, multiplex genome editing, functional genetic screen, expanded genetic library, uh, artificial genome synthesis, artificial gene network, recombinant vector, episomal vector, viral vector, shuttle vector, DNA assembly, Gibson assembly, uh, Golden Gate cloning, gateway cloning, uh, Buffalo virus system, viral expression system, sgRNA, guide RNA, uh, CRISPR interference, CRISPR activation, CRISPR screening, CRISPR learning, CRISPR library, okay, synthetic organism, synthetic promoter, metabolic engineering, pathway engineering, enzyme engineering, protein design, protein folding, molecular chaperone, artificial enzyme, synthetic chromosome engineering, synthetic metabolic pathway, directed metabolic evolution, direction evolution directed evolution experiment, DNA synthesis technology, synthetic nucleotides, artificial ribosomal, okay, genonucleic acids, lentiviral transduction, adeno-associated virus. Okay, I completed 50 genetic terms if you count. Okay, next, genetic biosensor, genetic circuit design, gene toggle switch, gene network modeling, uh, genome scale engineering, genome writing technology, genome scale knockout, gene delivery nanoparticle, gene therapy vector design. Okay, uh, targeted genome correction. Okay, next generation gene therapy, crop genome engineering, evolutionary protein engineering, rapid genome detection, pathogen genome sequencing, viral genome surveillance, agricultural gene editing, livestock genome editing, industrial biotechnology genetics, molecular memory storage, bioengineered pathway, bioengineered network, bioengineered micro, uh, biocomputing DNA, biological oscillator, biological feedback loop, engineered bacteriophage, synthetic cell, minimum cell, protocell model, DNA nanostructure, DNA origami, DNA logic gate, okay, DNA vaccine technology, RNA therapeutic, mRNA vaccine platform, okay, CRISPR base editor, CRISPR primate, CRISPR diagnostic test, CRISPR multiplex genome editing, okay, molecular storage, molecular nanotechnology, okay, synthetic synthetic gene circuit, okay, synthetic gene oscillator, okay, synthetic gene oscillator, come on, synthetic gene oscillator, synthetic virology, synthetic regulatory pathway, synthetic regulatory network, synthetic genome project, synthetic virus vector, synthetic vaccine platform, lipid nanoparticle system, DNA storage technology, directed evolution experiment. Okay, completed 50 terms, total 100. Genetic prediction model, genomic risk prediction, genome well prediction, genetic climate adaptation, genetic genomic printing, genetic nanorobotics, genome cloud database, genetic frontier science, gene drive ecology, genome design software, genome information compression, AI genomics, artificial intelligence genomics, machine learning genetics, deep learning genomics, predictive genomics, personalized genome medicine, digital genome archive, space genetics, astrobiology, biology genetics, quantum biology genetics, artificial life genetics, conservative genomics, de-extinction genetics, engineered ecosystem genetics, designer enzymes, designer genomes, next generation genomics, computational genome engineering, radiation genome engineering, living material engineering, robotics genome engineering, DNA and digital storage, environmental gene editing, future human genome editing, computational evolution, evolutionary synthetic biology, evolutionary algorithm, bioengineered evolution, artificial evolution simulation, digital organism evolution, polygenic risk molecular missions, synthetic biodiversity, synthetic species design, synthetic genome factory, synthetic S3 mobile genome, biofoundry systems, interplanetary biosystems, DNA computing, automated DNA synthesis, and molecular data recoding, encoding, auto agent genome resurrection. Okay, I completed again 50 times, 150 total. Genomic future medicine, genome reconstruction, genome resurrection, genome genetic planetary adaptation, genomic space exploration, genetic age reversal, genetic resilience engineering, genetic drought resistance, genetic ethics framework, genetic containment technology, genetic disease eradication, genomic immune engineering, genetic genome guided medicine, genome guided agriculture, astrogenomics, food security genomics, crop resilience, crop genome resilience, artificial genome ecosystems, regenerative genomics, universal vaccine genetics, synthetic gene circuits, synthetic antibody genes, synthetic developmental genetics, synthetic environmental microbes, bioengineered longevity genetics, bioengineered carbon capture genes, biosecurity genomics, biodiversity, biosecurity genome archive, cellular reprogramming genetics, cosmic radiation genome study, evolution monitoring genomics, climate adaptation genomics, ultra long DNA sequencing, DNA based computational systems, DNA repair enhancement, organ regeneration genes, induced pluripotent stem genetics, longevity gene pathways, telomere extension therapy, precision genome surgery, predictive evolution modeling, agriculture, artificial evolutionary intelligence, global genome surveillance, planetary genomic database, germline genome editing, embryonic genome correction, human genome redesign, aging genome modification, pathogen resistance engineering, adaptive immune genome editing. Okay, total 200 terms. Okay, next, genetic engineering civilization, genetic science, genetic engineering civilization, genetic civilization science, genetic planetary stewardship, genetic knowledge frontier, global genome mapping, global genome library, global genetic observatory, genome driven technology, genome scale biotechnology simulation, global genomic network, post human genomics, post genomic biology, hypergenomics, hyperscale genomics, future genetics, future of life genomics, next generation genetics, cosmic evolutionary genomics, uh, evolutionary megagenomics, artificial biosphere genetics. Okay. Next, future molecular genetics, interstellar biology genetics, biology genetics, synthetic ecosystem genetics, synthetic genome civilization, synthetic biological intelligence, synthetic evolutionary intelligence, synthetic genome societies, molecular evolution frontier, bioengineering future species, bioinformatics supercomputing, universal genetic language, universal genetic catalog, universal genome theory, universal life course study, ultimate genome engineering, infinite genome exploration, digital life code, digital genetic twin, DNA based artificial life, cosmic life genome theory. Okay. Predictive evolutionary simulation, artificial genetic network, evolutionary algorithm, evolutionary bioengineering, evolutionary system modeling, evolutionary genome simulation, planetary biosphere engineering, multi-species genome atlas, advanced genomic analytics, whole life genome tracking, AI assisted genome design, artificial intelligence assisted genome design. Total 250 terms completed. Okay. Next, total 5 categories. Sixth category, gene, DNA, RNA, chromosome, genome, allele, genotype, phenotype, heredity, trait, dominant, recessive, homozygous, heterozygous, locus. Okay. Next, nucleotide, double helix, adenine, thymine, cytosine, guanine, base pair, codon, anticodon, transcription, translation, replication, DNA polymerase, RNA polymerase, genetic code. Okay. Next, mitosis, meiosis, gamete, diploid, haploid, crossing over, recombination, sister chromatids, spinal fibers, centromere, Punnett square, law of dominance, law of segregation, law of independent assortment, monohybrid cross, dihybrid cross, test cross, back cross, probability, hybrid. Next, mutations and variation category, mutation, point mutation, deletion, duplication, insertion, inversion, frameshift mutation, silent mutation, missense mutation, nonsense mutation, polymorphism, single nucleotide polymorphism, SNP, genetic variation, genetic drift, gene flow, population genetics, population, allele frequency, natural selection, adaptation, fitness, bottleneck effect, founder effect, migration, evolution, speciation, human genetics and ancestry, autosomal DNA, Y-DNA, mtDNA, ancient DNA, haplogroup, genetic ancestry, genetic admixture, genetic lineage, genetic marker, genetic distance, phylogenetics, genome sequencing, human migration, population structure, molecular clock. Next, polymerase chain reaction, PCR, DNA sequencing, CRISPR, gene therapy, gene editing, genomics, genetic engineering, cloning, bioinformatics, proteomics. Next, gene activation, gene regulation, gene repression, operand, lack operand, regulatory gene, sister gene, regulatory sequence, DNA binding protein, okay, transcription factor, operator, terminator, start codon, stop codon, RNA splicing, alternative splicing, messenger RNA, transfer RNA, ribosomal RNA, non-coding RNA. Okay. Next, zygote, genetic lineage, epigenetics, genetic map, genetic disorder, genetic diversity, genetic recombination, genetic genome, gene expression, genome editing, DNA repair, mutation rate, admixture, human genome, epistasis, chromosomal aberration, polygenic trait, ribosome, transcriptomics, horizontal gene transfer, histone, telomere, chromatid, exon, intron, promoter, enhancer, oncogene, tumor suppressor. Okay. Next, chromatin, euchromatin, heterochromatin, histone modification, CpG alien, DNA methylation, satellite DNA, mini satellite, microsatellite, tandem repeat, karyotype, chromosomal deletion, chromosome duplication, chromosomal instability, chromosomal translocation, sister chromatid exchange, trisomy, monosomy, aneuploidy, polyploidy. Next, incomplete dominance, codominance, polygenic inheritance, mitochondrial inheritance, maternal inheritance, paternal inheritance, sex-linked inheritance, Y-linked inheritance, X-linked inheritance, imprinting, multiple alleles, pleiotropy, genetic penetrance, genetic predisposition, genetic mosaicism, career, variable expressivity, germline mutation, somatic mutation. Okay, next gene pool, genetic isolation, reproductive isolation, genetic bottleneck, genetic convergence, phylogenetic tree, evolutionary divergence, ancestral allele, derived allele, introgression, hybridization, adaptive mutation, neutral mutation, selection pressure, molecular evolution, founder population, stabilizing selection, directional selection, disruptive selection, whole genome sequencing, shotgun sequencing, DNA microarray, gene cloning, gene drive, genome assembly, genetic screening, pharmacogenomics, functional genomics, comparative genomics, restriction enzyme, leakage enzyme, plasmid, recombinant DNA, vector DNA, plasmid. Next, started first. Gene pool, genetic isolation, reproductive isolation, genetic bottleneck, genetic convergence, phylogenetic tree, evolutionary divergence, ancestral allele, derived allele, introgression, hybridization, adaptive mutation, neutral mutation, selection pressure, founder population, molecular evolution, stabilizing selection, stabilizing selection, directional selection, disruptive selection. Okay, next, whole genome sequencing, shotgun sequencing, DNA microarray, gene cloning, gene drive, genome assembly, genetic screening, pharmacogenomics, functional genomics, comparative genomics, recombinant DNA, vector DNA, plasmid, restriction enzyme, ligase enzyme, DNA fingerprinting, gel electrophoresis, synthetic biology, carrier testing, prenatal testing. Okay, cRNA, mRNA, RNA editing, RNA interference, promoter region, enhancer sequence, insular sequence, silencer sequence, transactivation, transrepression, post-transcriptional regulation, post-transcriptional modification, riboswitch, acylation, glycosylation, phosphorylation, gene silencing, cis-regulatory element, transregulatory factor, heat shock protein. Next, gene promoter mutation, reproductive mutation, gene doses, gene amplification, gene suppression, copy number variation, transcription initiation, signal peptide, nuclear localization signal, ubiquitination, proteasome, chaperone protein. Okay, gene network, system genetics, regulatory pathway, chromatin loop, chromatin remodeling, RNA degradation, mRNA stability, trans elongation, trans termination, Kozak sequence, translational control, DNA demethylation, histone ester transferase, histone deacetylase, epigenetic regulation, ribosomal binding site, Shine diagram sequence. Next, 18th. Next, topologically associating domain, chromosome arm, chromosome arm, P arm, Q arm, chromosome banding, G banding, C banding, chromosome rearrangement, karyogram, ring chromosome, isochromosome, chromosome fragmentation, chromosomal fusion, Robertsonian translocation, reciprocal translocation, telomerase, subtelomeric region, pericentromeric region, B chromosome, supernumerary chromosome, cell operation, genome instability, genome duplication, segmental duplication, replication origin, replication fork, replication bubble, single strand binding protein, leading strand, lagging strand, single strand primase, helicase, Okazaki fragment, DNA linkage, replication checkpoint, topoisomerase, homologous recombination, homologous non-homologous enjoining, DNA mismatch repair, DNA damage response, DNA proofreading, DNA strand break, DNA damage signaling, base excision repair, nucleotide excision repair, chromosomal segregation, spinal checkpoint, aneuploidy, Unemployed cell, polyethylene chromosome, Lamprey chromosome. Okay. Next, gene frequency, gene interrogation, genetic equilibrium, Hardy-Weinberg equilibrium, genetic Klein, genetic stratification, genetic divergence, genetic hitchhiking, parallel evolution, divergent evolution, convergent evolution, phylogeography, population mixture, population bottleneck, wonder lineage, evolutionary lineage, introgressive hybridization, purifying selection, positive selection, balancing selection, selective sweep, alike variation, fitness landscape, selection coefficient, mutation selection balance, adaptive radiation, neutral evolution, molecular adaptation. Okay. Next, adaptive ancestral population, derived population, evolutionary rate, substitution rate, pharmacogenomic analysis, synonymous substitution, non-synonymous substitution, genetic clock calibration, genetic bottleneck recovery, genetic drift variants, genetic cluster, evolutionary genetics, population expansion, population effective size, population differentiation, demographic history, deep ancestry, ancestry haplotype, ancestral haplotype. Okay. Fixation index, FST, lineage sorting, coalition theory, genetic coalitions. Okay. Genetic susceptibility, genetic counseling, genetic biomarker, genetic vaccine, DNA vaccine, RNA vaccine, genetic pharmacogenetics, genetic registry, genetic risk score, genetic epidemiology, gene delivery, genetic screening panel, genomic wide association study, clinical genomics, translational genetics, medical genomics, pedigree analysis, linkage analysis, precision medicine, personalized medicine, germline gene therapy, somatic gene therapy, X-linked disorder, Y-linked disorder, Mendelian disorder, automatic dominant disorder, automatic recessive disorder, viral vector, adenovirus vector, lentiviral vector, oncogenomics, genomic medicine, chromosomal abnormality, somatic mosaicism, tumor mutation, tumor suppressor gene, driver mutation, passenger mutation, familiar mutation, disease mutation, rare variant, multifactorial disease, carrier frequency, newborn screening, diagnostic gene test, modern diagnosis, disease penetrance, variant classification, pathogenic variant, therapeutic, gene editing. Okay. Genome project, human genome project, gene libraries, genome library, cDNA library, synthetic gene, synthetic genome, genome annotation, sequence alignment, structural genomics, metagenomics, epigenomics, metabolomics, multiomics, long range sequencing, short range sequencing, nanopore sequencing, single cell sequencing, next generation sequencing, bioinformatic pipeline, gene scaffolding, gene prediction, genomic variant calling, gene expression atlas, gene expression profiling, genetic database, genomic biomarker, genome algorithm, genome visualization, genome browser, genomic signature, genome assembly, genetic editing platform, genome editing platform, genetic engineering vector, genetic interaction network, population genomics, evolutionary genomics, miscellaneous genomics, computational genomics, proteomics, transcriptomics, compared to transcriptomics, functional genomics compared to genomics, sequence database, annotation pipeline, sequence synthetic biology circuit, functional annotation, variant annotation, structural variant detection. Okay. CRISPR-Cas9, CRISPR-Cas12, CRISPR-Cas13, base edit, prime edit, gene replacement, gene knockout, gene knock-in, gene toggle, gene circuit, genetic switch, genome minimization, minimal genome, gene drive system, population gene drive, genetic modeling, genotype phenotype map, genetic recoding, genetic containment, bio containment system, genome design, genome transplantation. Okay. Oncogenomics, optogenetics, future genomics, next generation genetics, reporter gene, predictive genomics, artificial cell, digital genome storage, DNA data storage, synthetic genobiology, computational biology, systems biology, protein engineering, evolutionary engineering, DNA nanotechnology, molecular assembly, green fluorescent protein, synthetic organism, artificial DNA base, expanded genetic code, dietary evolution, conditional knockout, synthetic promoter, synthetic chromosome, artificial chromosome, precision breeding, bioengineered organisms, evolutionary simulation, molecular evolution experiment, gene cluster, gene conversion, gene duplication event, tandem duplication, retrotransposition, DNA transposition, DNA recombinase. Okay. Next, mobile genetic element, transposable element, line element, sign element, integrase, accessionase, transposase, recombinase enzyme, allo sequence, site specific recombination hotspot, homology search, gene shuffling, RNA polymerase R1, RNA polymerase 2, RNA polymerase 3, RNA helicase, RNA binding protein, DNA binding domain, DNA looping, meiotic recombination, domain recombination, structural motif, Leukin zipper, chromatin adaptation, chromatin compaction, chromatin architecture, nuclear chromatin domain, DNA topology, DNA strand invention, holiday junction, resolution engine, next, okay, I think supercoiling, epigenetic memory, transcriptional bursting, multi-gene family, zinc finger protein, helix twin helix motif, break induced mutation, protein domain evolution, recombination intermediate, nucleosome positioning, epigenome, epimutations, epigenetic clock, epigenetic drift, epigenetic inheritance, epigenetic stability, epigenetic editing, epigenomic profiling, epigenomic regulation, environmental epigenetics, nutritional epigenetics, developmental epigenetics, genomic imprinting, germline epigenetics, somatic epigenetics, next, migration genetics, population genome analysis, population genomic dataset, ancestral or ancient genome sequencing. Okay. Next, CRISPR-Cas9. Okay. Sorry. Let's start first. Epigenome, epigenetic epimutations, epigenetic clock, epigenetic drift, epigenetic inheritance, epigenetic stability, epigenetic editing, epigenetic reprogramming, epigenomic profiling, epigenomic regulation, environmental epigenetics, developmental epigenetics, nutritional epigenetics, genome imprinting, germline epigenetics, somatic epigenetics, histone variation, histone phosphorylation, histone methylation, histone ubiquitination, histone code, histone writer proteins, histone writer enzymes, histone eraser enzymes, polycomb group proteins, heterorast group proteins, DNA methylase, DNA methylationase, DNA methyltransferase, DNA accessibility, parent of origin effect, DNA chromatin remodeling complex, chromatin insulator, chromatin compartment, active chromatin, repressor chromatin, chromatin marks, chromatin state map, chromatin interaction, chromatin immunoprecipitation, IC sequencing, attack sequence, chip sequence, CDCA protein, topology domain, boundary element, DNA hypersensitive site, nucleosome remodeling, chromosome conformation capture, CRISPR epigenome editing. Okay. Next, gene clustering, gene contact zone, gene genetic clustering, genetic contact zone, genetic diffusion, genetic ancestry inference, genetic admixture claim, genetic founder event, genetic isolation by distance, genetic similarity, genetic continuity, genetic replacement, genetic diversity index, genetic landscape, genetic differentiation, genetic contact region, genetic foundation lineage, founder lineage, genetic ancestry component, phylogenomics, anthropological genomics, archaeogenetics, principal component analysis genetics, human genetic diversity, ancient DNA analysis, haplogroup lineage, maternal lineage, paternal lineage, autosomal admixture, population bottleneck event, migration wave, identity by descent, identity by state, population migration, population structural analysis, admixture test, admixture graph, admixture graph, admixture wave, ancestor population split, human dispersal, demographic expansion, migration corridor, linkage disequilibrium, haplotype block, ancient population, ancestral genome, ancient lineage, Paleolithic, Neolithic, Bronze Age ancestries, recombination map, alike richness, population stratification, genetic anthropology. Next, effort to rate estimate. Next, genetic timeline, genetic or group, genetic interrogation, genetic continuity hypothesis, replacement hypothesis, genetic bottleneck recovery, genetic founder population, genetic isolation event, genetic isolation event, genetic ancestry map, genetic diversity hotspot, genetic signal, genomic migration signal, genetic drift signal, genetic expansion zone, genetic admixture test, genetic ancestry algorithm, next migration genetics, population genome analysis, population genomic sequencing dataset, ancestral genome sequencing, human genomic diversity, evolutionary migration model, ancient genome reconstruction, population history reconstruction, paleogenetic sample, early human migration, human paleogeography, Neanderthal ancestry, Denisovan ancestry, ancient admixture. Next, human phylogeny, haplotype sharing, ancient hybridization, archaic introgression, population divergence time, migration pulse, deep ancestry lineage, demographic inference, demographic simulation, evolutionary anthropology, human genetic sample or tree, ancestral haplogroup tree, population genetic tree, migration modeling, migration rate estimate, coefficient simulation, human dispersal route, ancestral recombination graph. Okay. Next, last and final, genome, transcriptome, proteome, haplotype, chimerism, mosaicism, dominant allele, recessive allele, alleic heterogeneity, heteroplasmy, Mendelian inheritance, non-Mendelian inheritance, splice set mutation, penetrance, amplification, allele, syntheny, amplification, autosome, syntheny, genome, transcriptome, proteome, haplotype, mosaicism, chimerism, alleic heterogeneity, heteroplasmy, dominant allele, recessive allele, Mendelian inheritance, non-Mendelian inheritance, splice set mutation, penetrance, amplification, autosome, cistron, fission, Syntheny, Dimi, Griffith, clean Griffith effect, deletion mapping. Fish means fluorescence in situ hybridization. NGS, new next generation sequencing. K, karyotyping. Okay, I completed more than 1000 genetic terms. If you count perfectly, I say 1002 genetic terms. Finally, I achieved this feat. This is not a normal video. I think in front of YouTube, in front of video, saying 1000 genetic terms, it is not a normal. This is page 1 only. There contains 10 pages. Road to 10,000 genetic terms. Okay, this is very big. Okay, not only genetic terms, I also remember genetic ancestries of particular countries. Okay, next time I complete genetic terms plus genetic countries ancestries, genetic ancestry countries. Okay, if you like my video, please like, share, subscribe to my channel and encourage me. Next time page 2, complete page 2. Okay, bye.